In this video, we're going to talk about the process of science and the scientific method. This lesson is in preparation for your Lab 1. I'm going to first talk about in terms of the history of the development of the scientific method. The human race didn't come up with the scientific method in just a few days. It's been a progression of scientists all throughout history who ended up trying different methods, philosophers, scientists, until the scientific method as we know it today was developed. Um, Now I'm going to show you something which is supposed to illustrate this fact, and also the fact that basically Google doesn't know everything. Um, just because you search for something and find it on Google doesn't mean that you should just stop there and accept everything that you see. So if you go to Google and search who first proposed the scientific method, the top result that you're going to get is going to be Francis Bacon. Francis Bacon has been credited with development of the scientific method during the Renaissance. However, that's not quite true. The First of all, as I mentioned, the scientific method has been developed over many years, but as we know it today, the hypothesis-based rigorous scientific method that we know was actually largely developed during what's called the golden age of Islam in the Muslim world in 11th century. So in 11th century, scientists Ibn al-Haytham, al-Biruni, and Ibn Sina, they were all scientists who contributed to the development of the scientific method as we know it today. So just a bit of a history for you. And again, these scientists didn't really wake up one day and come up with these methods. They tried, and through philosophy and experimentation, they tried different things, and finally, collectively, they came up with the ideas of the rigorous scientific method as we know it today. So then what happens in the 12th century, a British scientist, Grosseteste, I hope I'm pronouncing it right, they translated the work of these Muslim scientists and introduced their ideas into the Western world. So the Western world wasn't the place where it was and Francis Bacon weren't the originators of the scientific method. So always be skeptical. So in science, there are two different types of science in general that can be done. One is observation science, and that's the kind of science that involves collecting data, observing nature. It usually involves scientists studying a phenomena that they really cannot manipulate, but they can observe, collect lots of data, draw correlations, and come to grand conclusions. In this picture, you see Jane Goodall, who was a primatologist, and she basically spent her life observing primates, and she taught us a lot about primates, and probably played a big role in conserving them, preventing them from extinction, or trying to. Another kind of science is experimental science, where you actually manipulate, you set up experiments, you develop hypotheses, and you manipulate an experimental model. This is Barbara McClintock. She was a scientist who won a Nobel Prize for her work on jumping genes. She through her work, she was the first person to show the world that the genes that we have on the chromosomes, actually, they're not always stationary and staying where they are. They actually jump from chromosome to chromosome. So we're going to spend today in this video mostly focusing on experimental science. But everything starts with an observation, so we're going to go through the step-by-step method of the scientific method. So it all starts with observation. A curious scientist observes something and then they ask a question about that observation. Now, based on the previous knowledge about whatever phenomena they're observing, based on the work that scientists have done before and their own previous observations, they formulate a hypothesis about why something is happening. They formulate a hypothesis that is aimed to answer this question. Then based on that hypothesis, they make a prediction. And then they design an experiment, do an experiment to test their hypotheses and analyze results. And two things may happen. Either the results support the hypothesis, which is great, the scientist can go to the next step, or the hypothesis is not supported. So the scientist goes back to the drawing board and tries to formulate another hypothesis. Now, the results, most of the time, when results are negative, aren't published, but sometimes they are. So once the results have been collected and conclusions were made, then they are reported in terms of publications. Now, there are other things that you need to consider and be aware of in scientific investigation. So conclusions in science are always evidence-based. So when you draw a conclusion, it has to be based on evidence, has to be based on experimentation. So you can't just come up and make up something out of the blue. There's always has to be evidence. There has to be consensus within the scientific community before an idea takes shape. So the reports of experimental results that are published, first they have to go through a peer review process, meaning that when scientists find the results, they write it, they submit it, then the publisher submits that paper to other scientists to review it based on their expertise, and they can either accept the paper or they may outright reject it, or they may ask for additional experimentation. And this is a very rigorous process. If you ever run into a scientist who's really happy about their publication being accepted, that's because they probably went through years of work and hell to get that published. Another thing about scientific findings is that they have to be reproducible. So just because one scientist identifies based on the experiment, reports something, and even though they get it published, if other scientists cannot reproduce those results, that type of conclusion isn't really supported. And just through this process of being evidence-based, being based on consensus and reproducibility, throughout history, some rare individuals who wanted to collect fame by making up data, they were actually caught. And with science, you should always have a healthy skepticism. With conclusions, scientists are never dogmatic. They always are open to the possibility that there are some things that we don't know, or conditions that we have not tested. So the hypotheses are not written, the conclusions and hypotheses that are expected, that are supported by evidence, are not thought at final 100% unchangeable conclusions. That's why it's kind of the wrong terminology to use when we use the word prove for scientific investigation. We really cannot use the word prove 100% because there's always going to be exceptions. So as a scientist, we should always have an open mind and be accepting of conditions we've never thought before. Science is also very iterative. You never come up with the perfect conclusion, the perfect system, perfect way of treating something. It is based on repeating and tweaking and improving. So never, we always hear maybe in pop culture or movies that a scientist wakes up one day out of a dream and thinks of something and makes it happen. It never happens like that. There's much iterative process of improving and repeating and tweaking. Scientists often formulate a hypothesis in the form of an if and then statement. So I've shown you a kind of a breakdown of it here. So if there is an assumed cause and effect relationship, then that's the predicted outcome and the experiment. So in here, let's say you are trying to decide what it is when you make bread. What is it that causes the bread to rise? And one hypothesis is that yeast that you put into the mix is the component that causes the bread to rise. So the hypothesis would be if yeast causes bread to rise, then bread made without yeast should not rise. So I hope that example makes it clear what an if and then a hypothesis statement, how it is formulated. Now, just wanted to say a few words for the difference between hypotheses and theories. First of all, theory in regular language, the way we speak about it, is different from the way that word is used in science. On a regular everyday language, when we say, I have a theory, or when we use the word theory, it implies a hunch. It's not a very sure thing. But in science, that's actually the opposite is true. Once something is bestowed the label of a scientific theory, this is something for which there is a vast amount of evidence that supports that theory, for which there has been no theory to reject it. So theory of evolution is one of those scientific theories. You might hear some people say, well, evolution is just a scientific theory, it's just a theory. Well, no, the use of the word that word in science is different than everyday use of the language. The scientific theory of evolution, the reason we call it a scientific theory, because there has been every piece of evidence that we have seen supports the theory of evolution, and we haven't been able to really find any evidence to not to support it. Okay, so the difference between hypotheses and theories. Hypotheses, they are a narrow scope, and they deal with a specific situation, with a specific proposed explanation for a situation under specific conditions. So, for example, let's say that here's a scientist. There is a scientist who's, like, let's say, looked at pictures of plants under the microscope, images of plants under the microscope, and the scientist has seen that all plants are made of cells. So then the next hypothesis that the scientists make is that, well, let's say cats are living organisms. I bet if I take, let's say, a little bit of a skin of a cat or something, and I look at it under a microscope, the cat is also made of cells. Or a dog could be made of cells. Humans are made of cells. So these are individual hypotheses that are dealt with specific situations. And of course, you can go and test it. A hypothesis doesn't really have any value unless it is testable. But what about theory? Well, theory is really broad in scope, and it gives general explanation that applies across a wide range of situations, and they're also testable. And basically it's there is really nothing, no observation that would refute them. So if you think about this thing about cats are made of cells, dogs, humans, plants, virtually during the past few hundred years or thousands of years that scientists have been studying life, what they have seen at every instant is that everything is made of cells. So then that all of these observations over many, many years gave rise to the cell theory. Now that doesn't mean that we're guessing that living things are made of cells. This cell theory is a scientific theory based on all those observations. Cell theory says all living organisms are composed of one or more cells, and we have yet to see a living organisms that at the most basic level is not made of cells. So theory is not a hunch, and it's not a grown-up hypothesis. It's a collection of the results from lots of different experiments, from lots of hypotheses that were supported by experimental results. That's when you develop a scientific theory. So now let's plan an experiment. Let's talk about this supplement. There is a claim that there is the component in this supplement which has a whole bunch of other stuff, that there's claim that this alpha lipoic acid in this supplement can help reduce blood sugar level. Now, when designing an experiment, there are factors that are called variables. Variables are factors that change. And there are two different types of variables. One kind is the independent or experimental variable. They're basically the same thing. The independent or experimental variable is that factor that is purposely manipulated by the experimenter. So for example, in terms of our yeast and baking experiment, the independent or experimental variable is that yeast in the bread. So in this case, in our example, the experimental variable is alpha lipoic acid. The dependent variable is that thing that you measure. A dependent variable, its value, its state is dependent on the independent variable, and it responds to change in the independent variable. In our example is the blood glucose levels. In terms of the bread example, the rising of the bread, that's the dependent variable. So another important thing when designing experiments is to look back at previous work that has been done and ask, what do we already know about blood sugar level? What else is there out there that affects blood sugar level? What has past research findings teach us about effects of such factors on blood sugar? And this is again goes back to the important fact that science is never done in isolation. Science always builds on itself. So scientists are always, when they're producing results, they're helping the future scientists, and they also themselves benefited from the science that was done before. So when we do all this past research and inquiry into the past research that has been done, we may identify other factors that can influence blood sugar levels, and we have to be cognizant of them. And these factors are called confounding factors, which if you're not careful, if you don't take them into account, they can really mess up the results of your experiments. So our supplement is our experimental variable, which we're trying to measure their effect on blood sugar level. But what are the confounding factors? Age could have to do with it. Depending on what your age you are, that changes blood sugar level. Race, some people in different races, they are more susceptible to increase in blood sugar. the weight that a person has. If a woman has had gestational diabetes, they are more prone to having high sugar levels. High level of inactivity increases sugar levels, and really whether or not you have high sugar level, actually it's interesting, depends on how fat is distributed in your body, and also it depends on family history. So there are all these other factors that you need to consider when you do the experiment. So if you're not careful, then you don't really know what you're looking at in terms of what is it, which one of these is changing blood sugar level. So the supplement that we're trying is the experimental variable is that alpha-lipoic acid in the supplement, and then these confounding factors, they need to be considered in the experiment, and the way you consider them, you check for them, is to you control them. That's why we call them controlled variables. So when we do an experiment, when we design the experiment, we want to make sure that if we're comparing two groups of people, one group takes the supplement, the other one doesn't, we want to make sure the individuals who participate in these two groups, they are from the same age, race, weight, gestational diabetes, and because we want to control for them. We don't want those factors to contribute to the results of the experiment. And we know all of this from the background research, all these factors. Another important factor to consider is that you want to eliminate the possibility that are due to chance. I don't think we realize how much chance has to do with the phenomena that we observe, and sometimes it's really surprising. One of the ways that we can guard against the occurrence of chance is to use large samples. So sample size should always be large. So for example, if you hear, let's say your friend takes this supplement and they say their blood sugar level goes down, well, does that mean that the supplement is making a change, or is that's just one person and really a proper experiment hasn't been done? And maybe that effect of blood sugar level was due to chance. So one of the ways to eliminate the effect of chance is by using large sample sizes, and also assigning subjects to experimental and control groups by random. So let's say if you enroll participants into a study, and you take those people that are of the same age, all those different factors, same age, race, weight, whatever. Let's say you get a whole bunch of them and you divide them into two, but you don't divide them based on particular criteria. You just divide them randomly, so that reduces the effect of chance. Now, another thing that you have to consider if you're a good scientist is to be careful of bias by the scientist and errors by subjects due to the placebo effect. So how do scientists control for that? Now you may be wondering, well, why would a scientist have bias? Well, sometimes we are human beings, and sometimes we can talk ourselves into believing something and seeing something. So knowing that, scientists guard against that, and I'll show you how they do that. And you've probably heard of the placebo effect, where you see an effect as a result of taking something that somebody gives you, but it really isn't, the result that you're seeing isn't because that drug or pill or supplement or whatever you're taking is responsible for it. Maybe the state of your mind changes and you took that placebo and you think you're taking something that is reducing your blood sugar level, and you go and exercise more, and maybe eat healthier, you become motivated to become healthy. So how do you deal with that? First of all, the placebo effect is controlled, checked for by doing single blind studies. That means neither the experimental group nor the control group know what they're taking. They don't know if they're taking the drug or the placebo. And then to address bias by scientists, the double-blind study is done, in which the scientist doesn't even know who is getting what. And usually there are companies that provide services where scientists hire them to conduct these experiments. So they don't even know who's getting what. So another thing is, okay, what is a proper placebo? And it really depends on how you're administering the experimental variable. If it's a pill, then the placebo should also be a pill that is missing that particular experimental variable that you're testing. If you are injecting a liquid that has that active ingredient in it, the placebo would treat the control group exactly the same way, except it would be missing that active ingredient. If it's inhaling, again, that device that the person control group inhales would be missing the active ingredient. So a proper placebo is not nothing. So you don't say, okay, we don't give anything to the control group. And it's not a sugar pill. I always very often hear a sugar pill as an answer, but a sugar pill isn't the answer, because there is no guarantee that the pill that had experimental control in it had sugar in it. So it has to be very, very specific. A proper placebo uses the same delivery method as the experimental variable, and it has everything but the experimental variable in it. So after watching this, I think you are now ready to complete the Lab 1 assignment.